All right, so in this video, I'll be discussing cellular respiration. Now, before we get into cellular respiration, I just want to review photosynthesis and how it relates to cellular respiration. So as you'll see, photosynthesis involves particular reactants, and then it produces certain products. The reactants for photosynthesis include 6 moles of carbon dioxide, 6 moles of water, and also energy from sunlight. The products of this reaction include 1 mole of glucose and 6 moles of oxygen gas. Essentially, plants utilize carbon dioxide, water, and sunlight in order to produce sugar and oxygen. So it is very obvious here that this reaction consumes energy. Because this reaction of photosynthesis consumes energy, it is termed an endergonic reaction. You will also notice that the photosynthesis reaction takes simple molecules and it builds complex molecules, such as glucose. When a reaction takes simple molecules and builds complicated molecules, this is termed an anabolic pathway. This is synonymous with anabolism. The reason I'm mentioning the type of reaction and pathway involved in photosynthesis is because all of these descriptors become flipped once we discuss cell respiration. So what is the definition of photosynthesis? This is a process which converts light energy to chemical energy. The light energy comes from sunlight, while the products of this reaction contain chemical energy. And as previously mentioned, photosynthesis is an anabolic pathway. This is because it builds complex molecules from simple molecules. It is essentially producing sugar and oxygen gas from carbon dioxide, water, and sunlight. Because photosynthesis must consume energy in order to perform work, it is termed an endergonic reaction. The chemical energy is stored in organic compounds. So an example of an organic compound is glucose. Plants can also synthesize or make fructose and sucrose through photosynthesis. These are all organic molecules because they are rich in carbons, hydrogens, and oxygens. Photosynthesis typically occurs in plants. It can also occur in algae and certain prokaryotic organisms. For example, cyanobacteria, which is a type of blue-green algae, is photosynthetic. Photosynthesis and cell respiration are sometimes described as having a cyclic relationship. Plant cells and animal cells are constantly undergoing their respective processes, and these processes don't occur one after the other, they actually occur simultaneously. So this cyclic description arises from the following relationship. Products of photosynthesis are used as reactants in cell respiration. And likewise, products of cell respiration are used as reactants in photosynthesis. These processes work simultaneously, that's why they are referred to as being cyclic. And one more note about photosynthesis. Photosynthesis does encompass the light reactions as well as the dark reactions, which are actually termed the Calvin cycle. So moving on to cell respiration. So now, this is very similar to the previous reaction as it has particular reactants and it produces certain products. So the reactants for cell respiration include 1 mole of glucose and 6 moles of oxygen, and these are required for the reaction. The products for cell respiration include 6 moles of carbon dioxide, 6 moles of water, and energy in the form of ATP. ATP is an acronym for adenosine triphosphate, which is an energy-rich molecule that our cells use to perform work. And eukaryotic cells use ATP in order to perform cellular functions and essentially cellular work. And just as a side note, I do mention cell respiration in the form of aerobic cell respiration because eukaryotes mostly utilize aerobic cell respiration, although they can also initiate anaerobic cell respiration. It is very obvious here, though, that this reaction releases energy, and so it is termed an exergonic reaction due to the net release of free energy. And you'll also notice that cell respiration takes a complex molecule and it breaks it down into simpler molecules such as carbon dioxide and water. And when a reaction takes complex molecules and breaks them down into simpler molecules, this is termed a catabolic pathway, and this is synonymous with catabolism. And so before I move on, I just want to highlight the importance of anabolic pathways and catabolic pathways. Together, all of the anabolic and catabolic pathways within an organism do make up the entirety of the metabolism of that organism. So what is the definition of cell respiration? This is a process which leads to the breakdown of organic molecules in order to produce energy. Because cell respiration takes complex molecules and breaks them down into simpler molecules, it is known as a catabolic pathway. And more importantly, this process does release energy for us to use in cellular functions and in general cellular work. Since this process releases energy, it is an exergonic reaction, as I previously mentioned. And so endergonic and exergonic reactions, these have to do with whether energy is consumed or released. These terms are related to energy, whereas catabolic and anabolic pathways have to do with whether complex molecules are broken down or if they are built up. And catabolic and anabolic pathways, as I previously mentioned, are related to metabolism. And so there's way more information to discuss about cellular respiration. It can occur in the presence of oxygen or in the absence of oxygen. So when cell respiration occurs with oxygen, it is termed aerobic cell respiration. And when cell respiration occurs without oxygen, it is termed anaerobic cell respiration. Aerobic cell respiration requires 3 detailed stages. In our textbook, Campbell Biology, the term cell respiration is used to describe the aerobic process. Therefore, cell respiration and aerobic cell respiration are used synonymously in the textbook. In aerobic cell respiration, oxygen acts as the final electron acceptor during a stage known as the electron transport chain, and in anaerobic conditions, other electron acceptors that are not oxygen are used. And just to review, the products of photosynthesis are used as the reactants in cell respiration. And likewise, the products of cell respiration are used as the reactants in photosynthesis, and these processes do work simultaneously. So in the next slide, we can see both of the equations for photosynthesis and cell respirations, and you may recognize some similarities. That is, the reactants in photosynthesis arise from the products of cell respiration. And likewise, you see that the reactants in cell respiration arise from the products of photosynthesis. And to review once more, photosynthesis is an endergonic reaction as it consumes energy. Not only that, photosynthesis maintains an anabolic pathway as it takes simpler molecules and builds more complex molecules. Cell respiration, on the other hand, is an exergonic reaction because it releases energy. And not only that, it also maintains a catabolic pathway as it takes complex molecules and breaks them down into simpler molecules. Now, I would like to add a new descriptor for these reactions. Both photosynthesis and cellular respiration are known as redox reactions. So before I discuss redox reactions, I just want to briefly illustrate what is really going on in photosynthesis and cell respiration. So here we have an animal which inhales oxygen and consumes sugar molecules. This provides the animal with energy. This animal will release water, and it will also release carbon dioxide into the atmosphere. And there is a plant standing nearby which will take in that carbon dioxide and also water from the atmosphere in order to produce sugar molecules and oxygen. In other words, photosynthesis and cell respiration are two sides of the same coin. Now, we'll discuss redox reactions. So redox reactions are one type of many types of chemical reactions. In particular, redox reactions involve the transfer of electrons from one atom to another or from one molecule to another. The term redox implies oxidation and reduction. So electron loss is known as oxidation. When you lose an electron, you become oxidized and you also act as a reducing agent. This is because you are reducing another atom and meanwhile becoming oxidized while doing so. Losing electrons contributes to the increased oxidation of an atom, sometimes described as the oxidation state. And so what do I mean by this? So I will give some examples. So for instance, an atom may go from a 3- charge to a 2- charge. Therefore, losing an electron increases the oxidation of that atom. The final charge may still be negative, but it is closer to a positive charge than it once was, hence the term oxidation. As another example, an atom may go from a 1- charge to no charge. The final charge may be neutral, but it is closer to a positive charge than it once was, hence the term oxidation. And I'll give a final example regarding oxidation. So an atom may go from a 1+ charge to a 2+ charge. The final charge is positive due to electron loss, again, hence the term oxidation. So essentially, electron loss will shift the charge of an atom towards the positive, regardless of whether the final charge is negative, neutral, or positive. It shifts it. So this means there is an increase in oxidation state. Now, electron gain, which is the opposite, is known as reduction. When you gain an electron, you become reduced, and you also act as an oxidizing agent. This is because you were oxidizing another atom and meanwhile becoming reduced while doing so. So gaining electrons contributes to the decreased oxidation of an atom. And I'll give some examples regarding reduction. So for instance, an atom may go from a 2- charge down to a 3- charge, and therefore gaining an electron decreases the oxidation of that atom. The final charge may still be negative, but it is further from the positive than it once was. Hence the term reduction. And then in another example, there may be an atom that goes from no charge down to a 1- charge. The final charge is negative, but that's irrelevant. It has gone down. It is further from a positive charge than it once was, hence the term reduction. And then in a final example, there may be an atom that has a 2+ charge and it goes down to a 1+ charge by gaining an electron. Even though the final charge is still positive, an electron was still gained, hence the term reduction. So reduction will contribute to the decreased oxidation of an atom. And again, just by gaining an electron, this will shift the charge of an atom towards the negative. And this means essentially there is a decrease in oxidation state. So now we're going to move on to the reducing and oxidizing agents. And as I've mentioned, an atom that becomes oxidized is itself a reducing agent. This is because it donates electrons. In other words, the reducing agent is an electron donor, which allows itself to become oxidized by giving away electrons. You might notice the text, the reducing agent directly reduces the oxidizing agent, and that may become a bit confusing. However, we can think of it as this. The electron donor directly reduces the electron acceptor by donating negative electrons to the electron acceptor. An atom that becomes reduced is itself an oxidizing agent. This is because it gains electrons. In other words, the oxidizing agent is an electron acceptor, which allows itself to become reduced by accepting negative electrons. And again, you notice the text, the oxidizing agent directly oxidizes the reducing agent. And again, that might be a bit confusing. However, think of it like the electron acceptor directly oxidizes the electron donor by accepting those negative electrons from the donor. And so electron acceptors are always reduced and electron donors are always oxidized. And so to build on this, charged atoms are termed as ions. So a positively charged atom is termed a cation specifically, and then a negatively charged atom is termed an anion. So ions arise from either the gain or loss of electrons. And then collectively, oxidation and reduction reactions, we term those as redox reactions. And to build upon this a bit further, redox reactions, they can involve a complete transfer of electrons, or they can involve just a partial transfer of electrons. So in this diagram, they have outlined the common charges for certain ions, and take a look at each element on this diagram. The number just above the element symbol will signify how many protons are in that atom. And recall that the number of protons is equal to the number of electrons. So let's take an example. We'll use sodium as an example. I have here elemental sodium and also elemental chlorine. The element of sodium contains 11 protons per that one atom. So therefore, we also have electrons, in the electron cloud within this atom, and 11 electrons exist within that one atom of sodium. The electrons, as I mentioned, exist in a cloud which surrounds the protons and neutrons. So electrons are typically drawn in rings, which are formally called shells. So these are, examples of shells. You'll notice that some atoms may just need one shell, and some atoms need two shells, and then some atoms, as I've drawn here, need 3 shells. When I say atom, I mean a different atom of a different element. So for example, sodium, chlorine, fluorine, etc. And so there are a few rules when inserting certain electrons onto these rings to try to visualize that electron cloud. So the first shell can house only 2 electrons, a maximum of 2 electrons. And remember, I only have 11 electrons here to work with regarding sodium. So once I place 2 electrons in the first shell, that is the maximum, all subsequent shells can house up to 8 electrons. And that's for all the elements. So you'll see in the second shell, I have inputted 8 electrons. And so at this time, I have placed 10 electrons and I have 1 remaining. So the 11th electron will be placed on that third shell. And this gives us a total of the 11 electrons as depicted by the periodic table. So something interesting you should know is that the outermost shell is known as a valence shell. And so the outermost shell of sodium just so happens to have just 1 electron within it. And while elemental sodium has no charge and is neutral, if sodium tries to bond to another atom, it will donate its electron, and then that makes it become oxidized, and then it becomes Na+. And therefore, the charge of a sodium ion is Na+, or 1+. So let's look at another example. Let's look at elemental chlorine. And chlorine has 17 electrons because it has 17 protons. So the first shell, as you know, can only contain a max of 2 electrons. And the second shell, I've inputted 8 electrons. And the third shell contains 7 because only 7 electrons are remaining. And this gives me a total of 17 electrons as depicted by the periodic table. And so in the valence shell, in the outermost shell of chlorine, I have 7 electrons. This will be important in the next slide when I discuss the bonding of sodium and chlorine. So let's move on to the next slide. Now, the redox reaction of sodium chloride, also known as table salt, is a bit interesting. Although sodium and chlorine in their elemental form are neutral, if they try to bind to another atom, such as each other, they one will have to donate an electron so that the other one can accept it and become reduced. So what occurs in this case is that sodium is the reducing agent because it will donate an electron to chlorine. And so when it donates an electron to chlorine, it is reducing chlorine. Na acts as the reducing agent because it is reducing chlorine. And since chlorine accepts that electron, it gladly accepts that electron, Na becomes oxidized. All right, so we can see that here. Na loses an electron and it becomes positively charged, it becomes oxidized. Chlorine gains an electron and it becomes negatively charged. It is now termed the chloride ion. And so in this case, chlorine is actually the oxidizing agent as it itself becomes reduced while also oxidizing sodium. So what will happen here is the electron is donated, chlorine accepts it, but it's one more electron than it is used to, so it now maintains a negative charge, and chlorine has lost an electron. It's become a bit oxidized, and it will have a 1+ charge. And for these two ions specifically, because they do make a salt, a plus charge and a minus charge will balance out, they will neutralize, and this will cause a neutral compound, also known as an ionic compound. And most of these salt compounds, which can be chlorides or sulfates, they are ionic. They are bound with ionic bonds, so they must have a neutral net zero charge. And for example, if you're interested, examples of salts include like sodium chloride, barium chloride, calcium chloride, sodium sulfate, magnesium sulfate, etc. So these all have net zero charges, and they are all ionic compounds held together by ionic bonds. So the reason I mention this is because going forward, it's important to recognize that photosynthesis and cell respiration are both actually redox reactions, and each of those respective processes, they actually contain smaller redox reactions within them and within all their stages. And so with that knowledge, we'll go forward to discuss cell respiration. So cell respiration or aerobic cell respiration, this includes 3 stages. And the first stage is glycolysis. The second stage is the Krebs cycle. It is also termed the citric acid cycle or TCA cycle. It can also be called the tricarboxylic acid cycle in some literature. And then the third stage is oxidative phosphorylation. You may have learned of the third stage as being termed the electron transport chain. In our textbook, Chemical Biology, the third stage is formally termed oxidative phosphorylation because it includes the electron transport chain as well as a very necessary process known as chemiosmosis. So in this chart, I've just outlined where aerobic cell respiration occurs and where anaerobic cell respiration occurs. So to review, we have eukaryotic cells and prokaryotic cells. So eukaryotic cells reside within plants, animals, fungi, and protists. These are cells from the domain Eukarya, which encompass the 4 kingdoms of Plantae, Animalia, fungi, and Protista. And in these eukaryotic cells, aerobic cell respiration begins in the cytosol, which is the semi-fluid portion of the cytoplasm. And then cell respiration then moves into the mitochondria or the mitochondrial matrix, and then it ends in the inner membrane folds of the mitochondria. And so the third stage, as I mentioned before, is oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. And so something to take away from this slide is that eukaryotic cells engage in aerobic cell respiration. And so both plant and animal cells will engage in cell respiration. Plants can engage in cell respiration, whether it is light or dark. However, plants can only engage in photosynthesis during light. And then to review prokaryotic cells, these are cells within the domains bacteria and archaea, and aerobic cell respiration occurs in slightly different locations in prokaryotic cells. So glycolysis and the Krebs cycle will occur in the cytosol in prokaryotic cells, and then oxidative phosphorylation will occur in the plasma membrane. And then let's just briefly discuss anaerobic cell respiration. So anaerobic cell respiration can be anaerobic, or it could lead to fermentation. And there are two types of fermentation. There's alcoholic fermentation and lactic acid fermentation. And so these processes do not require oxygen, but they do require glycolysis. So glycolysis is actually the first stage in these processes. So jumping into glycolysis, and this relates to the first stage of cell respiration. The root words glyco and lysis literally mean to split sugar, or just the degradation of sugar. This is a 10-step cycle which functions in the initial splitting of glucose molecules. And let me emphasize that it is the initial splitting of a glucose molecule. As I previously mentioned, glycolysis occurs in the cytosol. This process is responsible for the breakdown of a 6-carbon sugar into 2 3-carbon molecules. And these resulting 3-carbon molecules are termed pyruvates. So for each glucose molecule, 2 pyruvates will result. And so in this slide, we can see the first 5 steps out of the total of 10 steps required for glycolysis. And you will notice that enzymes are required for each step. So we begin in glycolysis with a glucose molecule, and then we finish up glycolysis with a product of 2 pyruvate molecules. So these 10 steps are needed in order to initially split 1 molecule of glucose into 2 molecules of pyruvate. So next up is the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle. So this is the second stage of cellular respiration. So when the textbook mentions cell respiration, as I previously mentioned, it is referring to aerobic cell respiration. The Krebs cycle requires 8 steps in order to complete the breakdown of glucose molecules. So glucose is completely broken down in the Krebs cycle. And as I mentioned, although the initial splitting of glucose occurs in glycolysis, the complete breakdown of glucose occurs during the Krebs cycle. And in eukaryotic cells, the Krebs cycle occurs in the mitochondria, and typically this is in the mitochondrial matrix. Here, pyruvate will become oxidized into acetyl-CoA. Essentially, this is pyruvate oxidation, and upon the oxidation of pyruvate to acetyl-CoA, acetyl-CoA will then act as an entry compound. So acetyl coenzyme A is an entry compound used within the Krebs cycle. And if you're interested, acetyl-CoA is made up of a 2-carbon fragment of pyruvate attached to a coenzyme. And so the Krebs cycle essentially functions in the complete breakdown of glucose into carbon dioxide. And as seen in the previous chart, in prokaryotic cells, the Krebs cycle will occur in the cytosol. So between glycolysis and Krebs, there is pyruvate oxidation. And pyruvate oxidation takes that pyruvate molecule and it oxidizes it into acetyl-CoA, which is an entry compound. So technically, pyruvate oxidation is one of the first steps in the Krebs cycle prior to initiating the Krebs cycle. So in this slide, we can see all 8 steps for the Krebs cycle. And you will notice there are a lot of redox reactions taking place within this cycle. And so hence the importance of oxidation reduction reactions. And then I will mention one note. If you do search the Krebs cycle on the internet or in different textbooks, some images may depict 10 steps. However, those are more drawn out steps, whereas Campbell Biology does condense these steps into just 8. So, but they are essentially all of the same steps. So next we have oxidative phosphorylation. Now this is the 3rd stage of cell respiration. Oxidative phosphorylation includes the electron transport chain and chemiosmosis. Some information about the electron transport chain. The electron transport chain involves the movement of electrons down a sequence of redox reactions. And here membrane proteins act as the electron carriers. The electron transport chain, it's essentially a collection of molecules embedded in the inner membrane of the mitochondria in eukaryotic cells. And the folding of this inner membrane of the mitochondria is actually called cristae, C-R-I-S-T-A-E. So these cristae provide an increased surface area within the inner membrane folds of the mitochondria. And as previously mentioned, most components of the electron transport chain are proteins, so they do exist in complexes. And so during the electron transport chain, electron carriers alternate between reduced states and oxidized states as they accept electrons and donate electrons. And then each component of the electron transport chain becomes reduced when it accepts electrons from its uphill neighbor. And then each component of the electron transport chain then returns to its normally oxidized form by passing those electrons down to its downhill neighbor. And then ultimately, electrons will combine with molecular oxygen and hydrogen ions in order to form water. And so the electron transport chain is a stage which results in energy release, but it is initially in the form of ADP. So in eukaryotes, energy is stored in the mitochondria, as you already may know, but energy in the form of ADP, also known as adenosine diphosphate, needs to be reacted in a certain way in order to form ATP. So what ends up happening is ADP will bind to an inorganic phosphate in order to produce ATP. And this is done through various redox reactions of the electron transport chain. And so that is how we take adenosine diphosphate, we bind it with inorganic phosphate, and then we create ATP. So in that method, ATP is synthesized. And this process has a name. So this is termed oxidative phosphorylation. So oxidative phosphorylation occurs when adenosine diphosphate binds with an inorganic phosphate to produce adenosine triphosphate. And this is, again, through various redox reactions of the electron transport chain. So not surprisingly, this is an efficient method. So 90% of ATP is created via oxidative phosphorylation. Now, there is a term, chemiosmosis, that you should know about because it relates to oxidative phosphorylation. And this is a mechanism which uses stored energy in the form of a hydrogen ion gradient. And so chemiosmosis will drive cellular work, such as the creation of ATP, and therefore chemiosmosis is used in aerobic cell respiration. So pulling this all together, oxidative phosphorylation entails the electron transport chain as well as chemiosmosis. So essentially, in the electron transport chain, you have the pumping of protons or hydrogen ions, H+, that create a hydrogen ion gradient across the membrane. And then chemiosmosis will allow for ATP synthesis to be powered by the flow of these hydrogen ions back across the membrane. So therefore, when you take the electron transport chain and couple that with chemiosmosis, this collectively is called oxidative phosphorylation. Now, there's another term you should know about, which is substrate level phosphorylation. And this is a different process which occurs early on near glycolysis and the Krebs cycle, actually. And substrate level phosphorylation is responsible for forming a smaller amount of ATP early on. So this process occurs when an enzyme transfers a phosphate group from something that is not inorganic phosphate. So you'll take the ADP, adenosine diphosphate, and you will bind it to a phosphorylated intermediate in order to create ATP, or adenosine triphosphate. And so again, to repeat that, this process of substrate level phosphorylation occurs when an enzyme will transfer a phosphate group from a substrate molecule, and that substrate molecule is a phosphorylated intermediate, and it will transfer that to ADP, and then you will create ATP. And so rather than adding an inorganic phosphate like we do in oxidative phosphorylation, in substrate level phosphorylation, we add a phosphorylated intermediate from a substrate molecule, and that is substrate level phosphorylation. And if you're interested, the substrate molecule, it refers to an organic molecule which was generated as an intermediate during the catabolism of glucose. So in the next slide, we see the electron transport chain coupled with chemiosmosis. And collectively, these two processes are termed oxidative phosphorylation. So just to reiterate, the electron transport chain relates to actual transport of electrons and pumping of protons across a membrane, and this creates a gradient. And then in chemiosmosis, we have ATP being synthesized or made, and this is powered by the flow of hydrogen ions back across the membrane. And so what is the point of all of this? Initially, you have that glucose molecule invested, but you also have two ATP molecules that you must invest in order to get this process going. And so essentially in the investment phase at the very beginning of glycolysis, you do need to invest one molecule of glucose and two molecules of ATP, and that is per glucose. And so you would net at the end of glycolysis, and we're specifically discussing glycolysis here, you would net two pyruvate molecules, which you already know about, and two water molecules, and more importantly, two ATP molecules. And so the whole process here, the point of this is to generate a lot of ATP per glucose molecule. And so investing just one glucose and two ATP at the beginning of glycolysis, what is the fruition of our investment, right? So the ATP yield at the very end, glycolysis, the Krebs cycle, oxidative phosphorylation, which includes the electron transport chain, the total amount of ATP generated will vary per glucose. But for our case, in Campbell biology, it roughly estimates about 30 to 32 ATP being created per one molecule of glucose. So that is 2 ATP plus 2 ATP plus 26 ATP, that's about 30. And then, plus 28 would be 32. Now, different textbooks and different resources on the internet might approximate there are 34 ATP yielded or 38, but in our case for this textbook, it does mention 30 to 32 ATP per molecule of glucose. And keep in mind, we are not just working with one molecule of glucose at a time. Our cells undergo cell respiration for countless molecules of glucose. And so these examples are just narrowing it down to 1 mole of glucose to try to illustrate how these processes work. And so in the next slide, I'll just be discussing catabolism from food sources. And as you already know, glycolysis does require glucose to begin, right? And glucose is actually a sugar molecule. So something important to note is that there are 4 biological macromolecules which you should know about. And one of these macromolecules is a carbohydrate, which is another word for a sugar molecule. So carbs are essentially sugar molecules, and these can take the form of monosaccharides, disaccharides, or polysaccharides. And something else you should note is that a carbohydrate is a polymer, and the monomers which make up the carb are monosaccharides. So these are small units which link together, small monosaccharides link together, thus creating a polymer known as a carbohydrate. So many monomers do make up one polymer. And, based on this information, as you can see here, a macromolecule, another word for that is a polymer. And the 4 biological polymers, also known as macromolecules, these include carbs, lipids, proteins, and nucleic acids. And as previously mentioned, each of these polymers is comprised of monomers. So for instance, the monomers within a protein are termed amino acids. The monomers within nucleic acids are termed nucleotides. And the monomers of a lipid, there's actually 2 monomers within lipids. Those are glycerol and fatty acids. And then again, the monomer of a carb is a monosaccharide. So these are examples of biological macromolecules, also known as polymers, and then their respective monomers. Now, catabolism, as you recall, is a process which breaks down complex molecules into simpler molecules. For instance, breaking down a polymer into its monomers is a catabolic process or pathway. And then we have anabolic and catabolic pathways, which are both metabolic pathways. And these can either build up or break down a complex molecule. So collectively, all of the anabolic and catabolic reactions within an organism are known as its metabolism. So this next slide is regarding anaerobic respiration. Anaerobic respiration uses electron acceptors other than oxygen to accept electrons at the downhill portion of its electron transport chain. And it also starts off with glycolysis, actually. And glycolysis does not require oxygen. So this is actually the same for aerobic cell respiration and now also anaerobic cell respiration. To reiterate, glycolysis does not require oxygen in cell respiration at all, whether it is aerobic or anaerobic. So regarding the anaerobic conditions, either anaerobic respiration or fermentation can take place. In anaerobic respiration, an electron transport chain is present. However, the final electron acceptor is not oxygen in anaerobic cell respiration. And then fermentation, which is a type of anaerobic cell respiration, it's actually a catabolic process which generates a small amount of ATP from some organic molecules. Something to note about aerobic cell respiration, anaerobic cell respiration, and fermentation is that they all use glycolysis to oxidize glucose. However, they differ in their final electron acceptor and whether an electron transport chain is used. So for aerobic cell respiration and for anaerobic cell respiration, an electron transport chain is used. However, fermentation does not use an electron transport chain, and because of this, respiration yields way more ATP in respiration than it does in fermentation. And so, as I mentioned, fermentation is a catabolic process and it will only make a small amount of ATP. In the absence of oxygen, many cells do use fermentation to produce ATP by substrate level phosphorylation. And as mentioned, glycolysis is the first stage of fermentation. So there are two types of fermentation, actually. There's alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation uses glycolysis in order to take pyruvate and reduce it down to ethanol, which is also known as ethyl alcohol. And in alcoholic fermentation, we also have NAD+, which is regenerated so that carbon dioxide can be released. And in lactic acid fermentation, pyruvate is still reduced. However, NAD+ is actually produced without having to release carbon dioxide. And then two of the common products formed from fermentation, as I've mentioned, are alcohol in the form of ethanol and lactate, the ionized form of lactic acid. And so in this diagram, I have the biochemical pathways involved in alcoholic fermentation and lactic acid fermentation. And then in the next slide, I have some terminology that is relevant, but these are terms that I could not fit in the previous slides. So these are terms relating to ATP, what a catalyst is, the Calvin cycle, which is the second stage in photosynthesis, and then other terms regarding plant cells and things like that. And then I also have a second slide with some continued terms that are important. However, I could not actually fit in the previous slides. So just as an overview, during glycolysis, each glucose molecule is broken down into 2 molecules of pyruvate. And then in eukaryotic cells, pyruvate then enters the mitochondria. In the mitochondria, pyruvate is oxidized into acetyl-CoA, also known as acetyl coenzyme A. And then during the Krebs cycle, also known as the citric acid cycle, acetyl-CoA will further be oxidized into carbon dioxide. And then here the electron carriers NADH and FADH2 will transfer electrons, which were derived from glucose, to the electron transport chain. And then during oxidative phosphorylation, the electron transport chains will convert this chemical energy into a more usable form, which can later be utilized for ATP synthesis. And this mechanism, which utilizes stored energy in the form of a hydrogen ion gradient, is termed chemiosmosis. Together, the electron transport chain and chemiosmosis make up the third stage of cell respiration, which is termed oxidative phosphorylation. And then during earlier steps of cell respiration, a few molecules of ATP are synthesized in a process called substrate level phosphorylation. So that concludes this presentation on cellular respiration.